So I'm going to talk about work that we recently published on how we're developing new single molecule techniques that allow us to probe directly structure-function relationships in molecular motors. All right. So, as we know, a very important idea in molecular biology is the relationship between three-dimensional structure and function. Unfortunately, most techniques that are available to us don't access that relationship directly. On one hand, you have structural data from X-ray crystallography that can give us some incredible detail, the 3D organization of the atoms, every atom in this protein. Here I'm showing two different structures of the same protein, the helicase, that's going to be the central player in today's story. And so you can learn about the different conformational states of the protein, but what you don't get is dynamics. You don't get function. Function has to be inferred. At the other end, you have all sorts of functional assays, bulk biochemical assays that can look at the unwinding activity of this protein. It's been extended to the single molecule assay. The single molecule fluorescence assays have been pioneered by the HA lab, and also single molecule assays based on force, magnetic waves that we heard about from Jan's talk, and optical traps. And these kinds of assays give you very detailed looks at the dynamics of these proteins. With very high sensitivity, you can look at the unwinding activity of a single helicase with base pair precision. But what you don't get out of this, most of the time, is information about the conformation of this protein that you're looking at. And so there's a disconnect between these two types of data sets. And our goal was to develop techniques where we can actually look at this relationship between the two directly. And so our approach to doing this, when I say we, this is a long-time collaboration between my lab and Taishin Ha's lab, is to combine the two workhorses of the single molecule field, which is single molecule fluorescence and force-based techniques, optical traps specifically. So TJ touched on this a little bit in his previous talk. Basically, a few years ago, we developed an instrument that consists of dual trap optical tweezers combined with a single molecule confocal microscope. And this instrument can allow us to measure both fluorescence and mechanical signals from the optical trap simultaneously. So as a proof of principle measurement, we did a very simple experiment. We essentially tethered a molecule of DNA that contains a small single-stranded DNA gap. And then in our fluidic chamber, we flow in oligonucleotide that's complementary to the single-stranded DNA region. And this oligonucleotide is labeled with a single dye molecule. And so if we raster scan our confocal spot specimen plane, occasionally you will see that one of these oligomers will hybridize, and you will see a bright spot between our beads. So if you park the confocal excitation spot right here in the middle, and you look at the time trace, you can see light turning on and off, corresponding to a single oligonucleotide hybridizing. So this basically demonstrates that we have single molecule fluorescence sensitivity. Now, simultaneously with this, we also get a mechanical signal, because when we get this hybridization reaction, we're turning single-strand DNA into double-strand DNA, which has different mechanical properties. And so you get a very slight mechanical signal that you measure simultaneously from the optical trap that's shown in blue here. And here, just to demonstrate the capabilities of the instrument, we actually picked a setup to give us a very small signal. We're actually capable of detecting signals that are sub-nanometers in size. So the key point is that this instrument allows us to measure single molecule fluorescence simultaneously with sub-nanometer mechanical measurements. So we're going to use this, fast forward a few years to today, to look at structure-function relationships in molecular motor. This is a helicase UVRD. It's a prototype for a large structural superfamily of helicases called a superfamily 1. It's one of these monomeric helicases that has two motor domains. These are basically inchworming along single-strand DNA using the energy of acute hydrolysis to translocate. And there are other accessory domains that I'll talk about in a second. So UVRD is involved in DNA repair processes like E. coli, two pathways specifically. One is nucleotide excision repair. UVRD is tasked with unwinding a small loop of DNA around the site of lesion, where then that strand can be excised and filled in by the repair polymerase. The second pathway is a methyl-directed mismatch repair, where UVRD has to basically unwind and a strand that has incorporated a mismatch due to replication errors. And it's involved in other functions too. So the reason why we got interested in UVRD in this class of enzymes is because there have been long-standing debates over its mechanism that have to basically related to structure-function relationship. So the first question is as follows. This is kind of the most basic question you can ask about conformation, which is how many helicases do you actually need to unwind it? Everyone agrees that a single helicase can translocate on single-stranded DNA, but there's disagreement over how many you actually need to unwind DNA. And so the crystal structure is basically indicated that a single helicase, a monomer, should be perfectly competent to unwind DNA based on the organization of the DNA around the protein, but there's a lot of biochemical and single molecular evidence that actually suggests that no, you need a dimer minimum to unwind DNA. And so we wanted to address this question, and so the way we approached it is by putting a fluorescent molecule tag on the proteins, and then we simply count how much fluorescence we have with the fluorescence part of our instrument, and use the optical trap to measure its unwinding activity. Okay? So with this, we worked with Tim Lohman at Washington University. They worked with this protein a lot, and they synthesized a protein that can be site-specifically labeled with a single dye molecule, so that we can use that to count. Okay, the second debate had to do with the two crystal structures that I showed you in the very first slide. It turns out that UVRD takes on two different conformations. These are known as the open and closed state. And the difference essentially is this accessory domain sign here, 2B, rotates these two structures by almost 180 degrees. And there the debate is over which of these conformations is actually active. So again, there are debates. The closed conformation was crystallized with DNA, and so those structural biologists argue that that must be the functional state of the protein. But again, biochemical evidence and some single molecular evidence suggests that actually it's the open state that is active. So which one is correct? To address this, we basically did a single molecule FRET experiment. We labeled these proteins with a donor-acceptor pair, so that in the closed state, these two dyes are close to each other. They give you a high FRET signal, and in the open state, gives you a low FRET signal. So the idea is to measure the FRET of this protein simultaneously with watching its unwinding activity with the trap, so we can actually correlate the two. Okay, so let's get this data. Oh, one thing I haven't said is how we actually measure unwinding with the optical trap. So the way we accomplish this is by tethering a DNA hairpin between our two beads. And we apply small force on this DNA, not enough to mechanically unwind the hairpin, but just to stretch the molecule out a little bit. And we maintain the system in a force feedback loop. We always maintain a constant tension in this molecule. Now we flow in our protein at ATP. And so as it starts to unwind the hairpin in front of it, we said to make up the slack, we have to maintain a constant tension. We have to move the optical traps further away from each other. And that becomes our readout for the unwinding activity. This delta X, how far we move the two traps away from each other. And we can measure this very sensitively. We can measure things at the scale of a single base pair of unwinding. Now, simultaneously with this, we have our confocal fluorescence, where we can now look at conformation, look at the number of proteins and also the conformational state. Okay, so here's the first type of experiment where we're going to use proteins that are labeled with a single dye molecule. We're going to count how many proteins are required for unwinding. This is a typical data trace that we measure. In red is the fluorescence that we measure. As you can see, fluorescence turning on in one step and going down in one step, indicating that we have a single dye labeled molecule loaded onto our DNA. At the same time, we're measuring the unwinding activity in blue here, and you can see that a monomer is basically capable of unwinding, because you see some activity there. But the activity is interesting because, first of all, you can see that the helicase doesn't make it very far into the hairpin, only about 12 to 15 base pairs that have a much longer helicase. And not only that, you see this back and forth motion where the helicase is unwinding, but you're also seeing the DNA re-zipping behind it, back and forth, back and forth, back and forth, until the protein dissociates. And so we call this kind of activity frustrated, because it's short range, it's repetitive, it's moving back and forth. Now, occasionally we get traces like this, where instead of the fluorescence dropping one step, it's actually two-step photobleaching, which tells us that we have two proteins loaded onto our DNA. You can see that the activity is very different. Instead of this repetitive back and forth motion, we get a much more gradual and long-distance unwinding trace, which we call long-distance unwinding. Okay, so if you take a histogram of all our traces and you just ask what's the maximum number of base pairs you unwind, you get a histogram like this, which clearly shows a steep drop-off around 15 or so base pairs that we basically use to bin our data into the two types of activities, these stereotypes of behaviors that we have, of frustrated and long-distance unwinding. Okay, so if we bin our data this way and then ask how many photobleaching steps we have in each population, you see this great correlation that frustrated activity is predominantly carried out by monomers, whereas the long-distance unwinding is done by dimers. So the answer to the first question is more nuanced than people had thought before. It's not that one is active and the other one's inactive. It's that you're getting two different types of unwinding activities from a monomer versus a dimer. So we got very interested in this, and we're really curious about these traces here of this repetitive back and forth unwinding and DNA zipping, and how a single helicase could actually be responsible for something like this. It turns out the answer we got was from doing the second type of experiment, where now we tag our protein with donor and acceptor pair and look at conformational dynamics within the protein between this closed and open state. Okay, so we repeated this measurement now in the conditions where we only loaded a single protein onto our DNA. This is this frustrated unwinding activity where you see unwinding, zipping, so forth, back and forth until the protein dissociates and you get a flat line. And now simultaneously with this, we're getting a donor and acceptor signals from protein, which we can turn into a FRET efficiency. Okay, so remember the FRET efficiency is basically telling us about the conformational state of the protein, whether it's in one state, the closed state, or the open state. So the first thing you notice is that the FRET efficiency is not constant. It's fluctuating. And it's fluctuating between two states. A low FRET state where the dyes are far apart, that's the open state, and a high FRET state that's the closed state where the dyes are close together. Okay, so as this thing is going along, it's actually switching back and forth between these two conformational states. So the beauty of this data is you can now correlate this bottom plot with the top plot. So to show you how this works, let me show in red shade time periods where it's in the closed state, and in green shade time periods where it's in the open state. So if you stare at this long enough, you will realize that whenever it's in the red state, the closed state, UVRD is usually going up. It's unwinding. Okay? And in the green state, the open state, is going down, which means DNA is rezipping. So, if we repeat this over many time intervals and many molecules, we can create a heat map. It's where the x-axis is the FRET state. This is our measurement of conformational states. The y-axis is the velocity. That's a measurement of function, where we define positive velocities as unwinding and negative velocities as DNA rezipping. So you can see this correlation carries out. The high FRET state or closed conformation of the protein, we get unwinding. And the low FRET state or open conformation of the protein, we get DNA rezipping. So again, the answer to the question is not active or inactive. It's actually related more to the directionality of motion, unwinding or DNA rezipping, which was really a big surprise to us. So how do we actually rationalize this kind of behavior? How is it possible that this helicase can go forwards or backwards? Well, the answer came to us from a number of sources. One is previous work done by the Coquette and Benson lab, where they proposed that UVRD and its homologs can do something called strand switching. Let me describe to you what I mean by strand switching. So UVRD translocates 3' to 5' along single-strand DNA. So if it's translocating along this strand right here, then that means it's moving towards the DNA fork in an unwinding direction. We could also imagine that this thing switches to the other strand, and while translocating 3' to 5', that would move it away from the fork, and that would allow the DNA to rezip in its wake. So that's precisely what we think is going on in this process. This back and forth motion is actually unwinding, strand switching, and then allowing the DNA to be zipped in the wake of the protein. That still doesn't answer why we see a correlation between this and the conformational state of the protein. For that, we can look at the crystal structures. And this 2D domain that's involved in this conformational switch, we see in the crystal structures, is actually contacting the duplex. It's basically acting like an anchor to the duplex. And in the closed state, what it does is it orients these motor domains so that translocation is towards the fork. So this would be the high fret state unwinding conformation. And during strand switching, what we think is going on is that this thing is switching to the open state and orienting these motor domains to go on the opposite strand so that it translocates away from the fork, allowing DNA to reset. So here's a movie of what this looks like. This is not a molecular dynamics movie, it's really just a rigid rotation. It's taking the crystal structure and doing rigid rotations of the individual domains. So we start off in the unwinding conformation high fret state, and now switching to the open conformation low fret state and rezipping configuration. I can't do dance like TJ does. Dot says I should try. Okay, so to conclude this part of the talk, the old model was that monomer versus dimer, closed and open, would really correspond to active versus inactive. But the answer is more nuanced than that. Monomer and dimer relates to these different types of activities, short distance, back and forth, and long distance. And the closed and open configurations actually correspond to orientations of the motor domains, either towards the fork or heading away from the fork, at least unwinding or rezipping. So we've discovered that these new features as a result of this combined technique. There's still a lot of open questions. For example, why is the dimer more processive than monomer? Why does it unwind more? Is there something to do with this conformational switch? That we don't know. We have some answers, but not all. So that's something that we're still looking into. Another question you can ask, well, if this is true, if you lock the helicase into a closed state or an open state, would you make it processive or non-processive? And the related question is this a general mechanism for these superfamily 1 helicases? And so I can actually answer these two questions here because while we were working on this technique, a graduate student in TJ's lab, Sinan Arslan, was doing some work on homologous EPRD, rep helicase, as the same issues of closed and open configurations. And what he did is he used a bifunctional crosslinker, or linker, to cross-link the protein into the closed conformation or the open configuration. So the closed one is called rep X, the open one is called rep Y. How am I doing on time? 3 minutes. Okay, good. I'm still going to skip over a lot of the work that he did and just focus on one experiment that we did in our lab, which is to put this in our optical trap assay and see what happens with these different cross-linked mutants. So what we find is that in the wild-type rep helicase and the one that's cross-linked in the open state, you get very little unwinding activity, but if you put in the one that is cross-linked in the closed state, you get a super helicase. You get something that can unwind very processively over 6 kilobase pairs. Basically, that was the length of our DNA tether that we fed to it, so it probably can do more. Not only that, but it's incredibly powerful. You can apply forces that resist unwinding up to 60 ppnewtons, and it can still unwind. So, that's amazing. Basically, just this confirmation. First of all, this is consistent with our previous result that the closed state puts it in the direction to unwind. And it's amazing that you can control the activity of these proteins just by this rotation of this domain. By the way, he was also able to do this with yet another homolog called PCRA, and you essentially get the same results. So this mechanism seems to be general to this class of proteins, which makes it interesting. So, the last minute I want to kind of speculate a little bit on how this might relate to the biological functions of these helicases. So, UVRD, rep, and PCRA involved in a whole assortment of different types of functions inside the cell. same protein is doing very different things depending on the context. And so what we think might be going on is, and all these different functions, they essentially require different properties, different orientations, different helicase processivities, and so forth. Just to give you an example of UVRD, I mentioned the two repair pathways that it's involved in. In nucleotide excision repair, UVRD is only tasked with unwinding a small bubble, about 10 to 20 base pairs per lesion. Contrast in mismatch repair, UVRD has to unwind up to a kilobase pair of DNA. That has to do with the specific mechanism by which this works. Very different requirements for the same protein. And so what we speculate is going on is that these different types of conformational states, the oligomeric state, but also this open and closed state, might be exploited by the cell or used by the cell to regulate the activities of these proteins so that they can perform different tasks at different times. And of course, this likely involves cellular targets like DNA binding proteins, but also things like SPL and so forth. So that's going to be a next direction for our lab to see how these different things are regulated. Okay, I think my time is up, so let me thank the people who worked on this. So the hero of this story was Matt Comstock, who was a CPLC postdoc that was shared between myself and Tachi Pa. He's now faculty at Michigan State, so he did all the work on developing this technique and the UVRD work. It's been taken over now by graduate student in my lab, Kevin Whitley. The cross-linking work was done by Sinan Arslan in CJ's lab, and he worked with my student, Stan Murphy's-off, to do the optical trap machines. Good. And we have our collaborators, Chris Thomas, Tim Blowman, and students, and sources of funding. With that, I will take questions.
Okay, are there any questions?
So your dimer is always bound there. It never goes out. You just change the orientation, it's moving.
The dimer, no, so we do see cases where you get this processive unwinding and then it'll switch non-processive unwinding. So there's certainly possibilities that the dimer can dissociate and you're left with a monomer that can't.
But can you actually do everything, just have the dimer fixed in there and just change the orientation between closed and open, or actually it has to come out?
So, okay, so one thing, we do not know in the dimer configuration what the state of the protein itself is, if it's closed or open. That's a good question. That's something we'd like to find out because we don't know why the dimer is more processive than the monomer. It could be that they interact specifically, it blocks the leading one into the closed state, for example, or they can be pushing each other non-specifically. So we don't know.
What I'm trying to figure out is basically these things bound all the time. They have to jump between one strand to the other strand, and then the open and close come. So what gives the fluctuation that you observe? Is that a random in and out between the two strands, or there is something controlling there?
There seems to be an effective sequence. So one thing we've noticed is that these reversals from one strand to the other seem to happen at a barrier in the hairpin where you get a strong GC. Talking about GC again. A strong GC region in the DNA. So we haven't quite worked out the mechanism, but one idea is that there's kind of a basal rate of strand switching, and if you get stuck at some barrier, basically, the pulse data may be kinetically.
But it appears that when you cross-link, you ignore all these things, you just keep moving forward.
Yeah.
All simply come details for the cross-link.
Okay, any further questions? Okay, then let's thank Jan one more time.